I actually was inspired by a relative of mine who ended up contracting acute leukemia, and it was at a time that was very little bit of therapy, that could be offered for patients with acute myelogenous leukemia, and just trying to study the options that were available, for this individual inspired me to look at hematological malignancies as a career. This is an indolent B-cell malignancy. It's classified by the World Health Organization as lymphoplasmacytic lymphoma. So when you hear, the term Waldenstroms, it's almost synonymous with lymphoplasmacytic lymphoma. The disease was first described by Jan Gosta Waldensstrom in 1943. He was a very famous Swedish doctor. He not only described Waldenstroms, but he also described carcinoid syndrome and porphyria, so other, important diseases. And, to do this all at the height of World War II was truly amazing. And it actually took about 15 years before his discovery was actually validated by others. So he took a lot of flack in those 15 years as to whether or not he had truly described, a unique disease. For the most part, our disease, Waldenstroms, has been the beneficiary of what I call hand-me-down therapies. We've had to wait until drugs were investigated in other disease groups, myeloma, CLL, lymphoma, before they could actually, be investigated in patients with Waldenstroms. And sometimes that lag could be many years. And so this has resulted because of the prioritization. Waldenstroms, has been classically thought of as an orphan, disease. But now recognizing really the statistics, it really isn't an orphan. I mean, there's a lot of patients who are diagnosed every year and who are living with Waldenstroms. And this is why, this, ability to bring Imbruvica, to these patients is such a, monumental feat. To think that 70 years went by that we had no approved therapy, and now we have, the opportunity to treat these patients with an FDA-approved drug. 3 years ago, we discovered the MYD88 mutation in Waldenstroms patients. This is a mutation that affects over 90% of people with Waldenstroms and has been validated now in over 30 studies. And in our laboratory, we wanted to understand, what this mutation, does. How does it affect Waldenstrom cells? And the key discovery that we published was that it activates Bruton's tyrosine kinase. Bruton's tyrosine kinase is the target of ibrutinib. And as we, worked on the science of this, we began to recognize what a, pivotal molecule Bruton's tyrosine kinase was for providing growth and survival cues to the Waldenstroms cells. I would like to thank my colleagues here at Pharmacyclics, as well as our colleagues at Janssen, for closely collaborating with us to be able to move Imbruvica forward in patients with Waldenstroms. Those close, meetings were very important, as we put together, the submission for a breakthrough designation, for the NDA filing. The ability to work very closely with the FDA was also very pivotal. And I think the recognition here is that this was really a team effort. And with this, I also want to thank, all our collaborators at Stanford University as well as at Memorial Sloan Kettering. Both their scientists as well as their clinicians played a pivotal role in being able to help us get this trial, to move forward. And lastly, I just want to thank all the patients, because a lot of these patients made great sacrifices, traveling in some cases thousands of miles, to come to their clinic appointments and to participate in this very important clinical study.